An evolution, accumulation of variation during reproduction. Inheritance from the previous generation provides both a common basic body design and subtle changes in it for the next generation. Now think about what would happen when this new generation in its turn reproduces. The second generation will have differences that they inherited from the first generation as well as newly created differences. Figure 9.1 would represent this situation if a single individual reproduces as happens in asexual reproduction. If one bacteria divides and then resultant two bacteria divides again, and the four individual bacteria generated would be very similar. There would be only very minor differences between them generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated, as we will see when we discuss the rules of inheritance. Do all the variations in a species have equal chances of surviving in the environment in which they find themselves? Obviously not. Depending on the nature of variation, different individuals would have different kinds of advantages. Bacteria that can withstand heat will survive better in a heat wave, as we have discussed earlier section of variants by environmental factors from the basis of evolutionary processes, as we will discuss in later sections. Heredity. The most obvious outcomes of reproductive process still remains the generation of individual of similar design. The rules of heredity determine the process by which traits and characters are reliably inherited. Let us take a closer look at these rules. Inherited traits. What exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human being. However, it does not look exactly like its parent, and human populations show a great deal of variations. Rule of for inheritance of traits. Mendel's contributions. The rule for inheritance of such traits in human beings are related to the fact that both the father and the mother contribute particularly equal amounts of genetic material to child. This means that each trait can be influenced by both parental and maternal DNA. Thus, for each trait, there will be two variations in each child. What will then the trait seen in the child be? Mendel's worked out the main rule for such inheritance, and it is interesting to look at some of his experiments for more than a century ago. George Johns Mendel. Mendel was educated in monastery and went on study science and mathematics at the University of Vienna. Failure in the examinations for teaching certificate did not suppress his zeal for science quest. He went back to his memory and started growing peas. Many others have studied that inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and mathematics and was the first one to keep count of individual exhibiting a particular trait in each generation. This helped him to arrive at the law of inheritance that we have discussed in the main text. Mendel used a number of consisting visible characters of garden peas, round wrinkled seeds, tall short plants, white violet flowers, and so on. He took pea plant with different characteristics, a tall plant and a short plant, produced progeny from them, and calculated the percentages of tall or short progeny. In the first place, there were no halfway characteristics in his first generation or F1 progeny, no medium height plants. All plants were tall. This meant that only one of parental traits were seen, not more mixture of the two. So the next question was, were the tall plants in F1 generation exactly the same as the tall plants of the parent generation? Mendelian experiments test this by getting both the parental plants and these F1 tall plants to reproduce by self-pollination. The progeny of the parental plant are of course all tall. However, the second generation or F2 progeny of the F1 tall plants are not all tall. Instead, one quarter of them are short. This indicates that both the tallness and the shortness traits were inherited in F1 plants, but only the tallness trait was expressed. Thus, two copies of the trait are inherited in each sexually reproducing organism. These two may be identical or may be different depending on the percentage. A pattern of inheritance can be worked out with this assumption as shown in figure 9.3. In this explanation, both TT and TT are tall plants, while only TT is a short plant. In other words, a single copy of T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits like T are called dominant traits, while those that behave like small t are called recessive traits. Work out with work out which trait would be considered dominant and which one recessive in figure 9.1 and 9.4. What happened when pea plants showing two different characters rather than just one are breed with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what would happen if F1 progeny are used to generate F2 progeny by self-pollination? A Mendelian experiment will find that some F2 progeny are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new mixtures. Some of them would be tall but have wrinkled seeds, while others would be short but have round seeds. Thus, the tall/short trait and round seed slash wrinkle seed traits are independently inherited. Another example is shown in figure 9.5. How do these traits get expressed? How does the mechanism of heredity works? Cellular DNA is the information source for making proteins in the cell. A section of DNA that provides information for one protein is called the gene for the protein. How do proteins control the characteristics that we are discussing here? Let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. Plant height can thus depend on the amount of particular plant hormone. The amount of the plant hormone made will depend on the efficiency of the process for making it. Consider now an enzyme that is important for this process. If this enzyme works efficiently, a lot of hormones will be made and the plant will be tall. If the gene for enzyme has an alteration that makes the enzyme less efficient, the amount of hormone will be less and plant will be short. Thus, genes control characteristics or traits. If the interpretations of Mendelian experiments we have been discussing are correct, then both parents must be contributing equally to the DNA of progeny during sexual reproduction. We have discussed this issue in the previous chapter. If both parents can help determine the trait in progeny, both parents must be contributing a copy of same gene. This means that each plant must have two sets of all genes, one inherited from each plant. For this mechanism to work, each germ cell must have only one gene set. How do germ cells make a single set of genes from the normal two copies that all the other cells in our body have? Progeny plants inherited a single whole genome set from each plant. Then the experiment in 9.5 cannot work. This is because the two characteristics R and Y would then be linked to each other and cannot be independently inherited. This is explained by the fact that each gene set is present not as single long thread of DNA but as separate independent pieces, each called a chromosome. Thus, each cell will have two copies of each chromosome, one each from the male and female plants. Every germ cell will take one chromosome from each pair, and these may have be either maternal or paternal origin. When two germ cells combine, they will restore the normal number of chromosomes in the progeny, ensuring the stability of DNA of the species. Such a mechanism in inheritance explains the results of Mendel experiments, and this used by all sexually reproducing organisms. But asexually reproducing organisms also follow similar rules of inheritance. Can we work out how their inheritance might work? We have discussed that the idea that the two sexes participating in sexual reproduction must be somewhat different from each other for a number of reasons. How is the sex of newborn individual determined? Different species use very different strategies for this. Some rely entirely on climatic clues. Thus, in some animals, the temperature at which fertilized eggs are kept determine whether the animal developing in the egg will be male or female. In other animals, such as snails, individual can change sex, indicating that sex is not genetically determined. However, in human beings, the sex of individual is largely genetically determined. In other words, the gene inherited from our parents decide whether we will be boys or girls. But so far, we have assumed that similar gene sets are inherited from both parents. If that is the case, how can we generate inheritance determine sex? The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and paternal copy, and we have 22 such pairs. But one pair, called the sex chromosome, is odd in not always being a perfect pair. Women have a pair of sex chromosomes, both called X, but men have a mismatched pair in which one is normal size X and while the other is a short one called Y. So women have XX and while men has XY. Now can we work out what the inheritance pattern of X and Y will be? As figure 9.6 shows, half the children will be boys and half will be girls. All children will have inherited an X chromosome from their mother regardless of whether they are boys or girls. Thus, the sex of child will be determined by what they inherited from their father. A child who inherits an X chromosome from her father will be a girl, and one who inherited Y chromosome from him will be a boy. Evaluation. We have noted that there is inbuilt tendency to variation during reproduction, both because of errors in DNA copying and such and as a result of sexual reproduction. Let us now look at some consequences of this tendency. An illustration. Consider a group of 12 red beetles. They live, let us assume, in some bushes with green leaves. Their population will grow by sexual reproduction and therefore can generate variations. Let us imagine all the crows eat these beetles. The more beetles the crow eat, the fewer beetles are available to reproduce. Now let us think about some different situations that can help in beetle population. In first situation, a color variation arises during reproduction so that there is one beetle that is green in color instead of red. This beetle, moreover, can pass the color on to its progeny so that all its progeny beetles are green. Crows cannot see green colored beetle on the green leaves of bushes, therefore cannot eat them. What happens then? The progeny of green beetle is not eaten while the progeny of red beetle continues to be eaten. As a result, there are more and more green beetles than red ones in the beetle population. In the second situation, again, a color variation arises during reproduction, but now it results in a beetle that is blue in color instead of red. This beetle can also pass the color on to its progeny so that all its progeny beetles are blue. Crows can see blue colored beetles in the green leaves of the bushes as well as they can see red ones, therefore can eat them. What happened initially in the population as it expands, there are fewer blue beetles, but most are red. But at this point, an elephant comes by and stumps on the bushes where the beetles live. This kills most of the beetles by chance. The few beetles that have survived are mostly blue. Beetle population slowly expands again, but now the beetles in the population are mostly blue. It's obvious that in both situations we started out as a rare variation came to be common characteristic in population. In other words, the frequency of inherited trait change over generations. Since genes control traits, we can say that the frequency of certain genes in a population changed over generations. This is the essence of the idea of evolution, but there are interesting differences too. But in the two situations, in the first case, the variation became common because it gave a survival advantage. In other words, it is naturally selected. We can see that the natural selection is exerted by the crows. The more crows there are, the more red beetles would be eaten, and the more proportion of green beetles in population would be. Thus, natural selection is directing evolution in beetle population. It results in adaptations in beetle population to fit their environment better. In second situation, colored change gave no survival advantage. Instead, it was simply a matter of accidental survival of beetle of one color that changed the common characteristic of the resultant population. The elephant would not have caused such major havoc in beetle population if the beetle population had been very large. So, accidents in small population can change the frequency of some genes in a population, even if they gave no survival advantage. This is the notion of genetic drift, which provides diversity without any adaptations. Now consider the third situation. In this, as the beetle population begins to expand, the bush starts suffering from plant diseases. The amount of leaf material for the beetles is reduced. The beetles are poorly nourished. As a result, the average weight of adult beetle decreases from what it used to be when leaves were plentiful. But there is no genetic change occurring. After a few years and a few beetle generations of such scarcity, the plant disease is eliminated. There is a lot of leaf food. At this time, what would we expect the weight of beetle to be? Acquired and inherited traits. We discussed the idea that the germ cells of sexually reproducing population are made in specialized reproductive tissues. If the weight of the beetle is reduced because of starvation, that will not change the DNA of the germ cell. Therefore, low weight is not a trait that can be inherited by progeny of a starving beetle. Therefore, even if some generation of beetle are low in weight because of starvation, that is not an example of evolution, since the change is not inherited over generations. Change in non reproductive tissues cannot be passed on to the DNA of germ cells. Therefore, the experiences of an individual during its lifetime cannot be passed on to its progeny and cannot direct evolution. Consider another example of how an individual cannot pass the on to its progeny the experiences of its lifetime. If we breed a group of mice, all the progeny have tails as expected. Now, if tails of these mice are removed by surgery in each generation, do these tailless mice have tailless progeny? The answer is no, and it makes sense because removal of tail cannot change the gene of the germ cells of the mice. Charles Robert Darwin. Charles Darwin set out on a voyage when he was 22 years old. The 5-year-old voyage took him to South America and the islands of its coast. The studies that he conducted during the voyage were to change forever the way we look at the variety of life on Earth. Interestingly, after he got back to England, he never left its shores again. He stayed at home and conducted various experiments that led him to formulate his hypothesis that evolution took place due to natural selection. He did not know that mechanism whereby variation arose in species. He would have been enlightened by Mendel's experiments, but these two gentlemen didn't know of each other or their work. We often associate Darwin solely with the theory of evolution, but he was an accomplished naturalist, and one of the studies he conducted was to do with the role of earthworms in soil fertility. This is the reason why the ideas of heredity and genetics that we have discussed earlier are so essential for understanding evolution. Even Charles Darwin, who came up with the idea of evolution of species by natural selection in 19th century, could not work out the mechanism. It is ironic that he could have done so if he had seen the significance of experiments his Australian contemporary George Mendel was doing. But then Mendel too did not notice Darwin's work as relevant to his origin of life on Earth. Darwin's theory of evolution tells us how life evolved from simple to more complex forms, and Mendel's experiments give us the mechanism for the inheritance of traits from one generation to next. But neither tell us anything about how life began on the earth in first place. JBS Haldane, a British scientist who became a citizen of India later, suggested in 1929 that life must have developed from simple inorganic molecules which were present on Earth soon after it formed. He expected that the conditions on Earth at that time, which were far from the conditions we see today, could have given rise to more complex organic molecules that were necessary for life. The first primitive organisms would arise from further chemical synthesis. How did these organic molecules arise? An answer was suggested by experiment conducted by Stanley L. Miller and Harold C. Urey in 1953. They assembled an atmosphere similar to that thought to exist on early Earth. This had molecules like ammonia, methane, and hydrogen sulfide, but no oxygen over water. This was maintained at a temperature just below 100 Celsius, and sparks were passed through the mixture of gases to simulate lightning. At the end of the week, 15% of carbon from methane has been converted to simple compounds of carbon, including amino acids, which make up protein molecules. So, can life arise afresh on Earth even now?